those digestive enzymes, and the acids will digest those molecules and break them into their monomers, and that's good for the cell. Occasionally, when cell biologists are looking at cells under microscopes, they see lysosomes not just fusing with food vacuoles, but fusing with some of the cell's own organelles. So sometimes these things eat some of the cell's own organelles, and that seems kind of puzzling. Nobody knows exactly what's going on there. Maybe it's just a mistake every now and then where the cell digests one of its own organelles, or it might be some sort of internal process that the cell does just to renew its own organelles. Maybe after a while the organelles start to get worn out, and so the cell wants to digest them up and start over again. Any lysosome questions? Okay. Y'all are kind of quiet today. I guess that's okay. Yes?
So no one really knows exactly why it is that they're breaking up?
Yeah, I mean, why the lysosomes digest some of the cell's own organelles, no one's 100% sure, but the leading theory is that just the organelles wear out after a while, so the cell digests them away to make new ones. Sort of internal recycling. Yes?
So there's no actual lysosomes floating around in the cell, they just kind of like put out from the Golgi whenever they're needed?
Yes, the Golgi manufactures lysosomes when needed, but if there's nothing that's supposed to be digested, the Golgi does not shoot out any lysosomes. They're made on a only when needed. All right, let's see. Another membranous organelle I want to discuss with you guys. Oh, go back. Here we go. Are structures called vacuoles. The vacuoles are storage sheds, I guess you'd say, inside the cell. They are hollow membranous spheres with little balls. The walls are made of phospholipid bilayer. And what's inside them is a watery environment where things are stored. What sort of things are inside there? Nutrients get stored inside vacuoles. Ions get stored inside vacuoles. In plant cells, pigments can be stored inside the vacuoles. Here's a close-up of a vacuole. And if it looks a lot like a vesicle to you, that's legitimate. Vesicles are little round balls of membrane that carry stuff. These are just larger balls of membrane with stuff inside. But the vacuoles aren't really for transportation. The vacuoles don't drift around inside the cell, transporting stuff from one spot to another. Vacuoles stay more or less stationary and just sort of hold the materials that they're supposed to be holding. Now, how does the vacuole get the material that it's supposed to be storing? Well, the answer there is vesicles. If there's something that needs to be stored in the vacuole, that material gets packaged in a vesicle like you see there, and then that vesicle then drifts through the cell and fuses with the vacuole and puts the materials inside the vacuole. Okay, now notice on the drawing there that this is an animal cell, and notice that we've been looking at vacuoles in there, so animal cells do have vacuoles, but plant cells have more vacuoles than animal cells do. And furthermore, plant cells have a special vacuole that animal cells don't have, and that special vacuole is called the central vacuole. And here's a picture of a plant cell, and that big thing in the middle is the central vacuole. So it's just a gigantic vacuole. In fact, it usually takes up most of the plant cell. Oftentimes in plant cells, the central vacuole is so huge, the other organelles, including the nucleus and all the other organelles in the Golgi body, get sort of smushed up against the side of the cell because of the gigantic central vacuole. But other than the fact that it's a huge vacuole, the central vacuole is Gesundheit, is still the same concept as all the other vacuoles that we see in animal cells and plant cells. It's a membranous sphere, It has water inside with typically dissolved nutrients and ions. Plant cells, in addition to nutrients and ions, also store some of their pigments. Pigments are colored molecules inside their central vacuole. If you think about some of the colors of leaves, especially in autumn time, you see a lot of yellows and oranges and reds. Those colored molecules are inside the plant's central vacuole. I remember reading something kind of interesting. Apparently, those pigments with the fall colors are always inside the leaf all year. And the reason why you don't normally see those colors during the growing season is because of the chloroplasts. These are the chloroplasts where photosynthesis takes place. And the chloroplasts have their own pigments. The chloroplasts have some green pigments. And there's so many chloroplasts, they essentially outnumber the pigments you see inside the central vacuole. And so that green essentially covers up the fall colors. But when fall comes, the chloroplasts die off, and that's when the more fall colors are revealed. All right, let's get back to the animal cell again for a second. So I've been going over these membranous organelles, remember what you find inside eukaryotic cells, but not prokaryotic cells. And let's do a quick wrap up here. There's, of course, the nucleus, endoplasmic reticulum, the Golgi apparatus, which is the sorting center, vesicles, the storage centers. I don't have it here, but a lysosome, which is the digestive organelle of the cell. All of these membranous organelles can exchange materials with each other by way of the vesicles. That is to say, vesicles are constantly going from, let's say, the endoplasmic reticulum to the Golgi, and then the Golgi sends its own vesicles out, some of which go to the nucleus, some of which goes to the vacuoles. The Golgi can send out lysosomes, which digest up food vesicles. The cell is really active and alive with vesicles transporting materials from here to there, to there, to there, to there, to there, to there, to there. And so all of these membranous organelles we've covered so far are really interconnected with each other. They exchange proteins and lipids and other materials with each other by sending or receiving vesicles. And for that reason, they are grouped together into something called the endomembrane system, which is all of these organelles that can exchange materials with each other. So that being said, there are three membranous organelles inside eukaryotic cells that are not part of the endomembrane system. So these three, which are peroxisomes, mitochondria, and chloroplasts, are membranous organelles, but they don't send vesicles, they don't receive vesicles. They are not part of the endomembrane system. Maybe a good way to analogy is free trade zones around the world. Like in Europe, for instance, there's various nations that freely send goods back and forth with each other. But there are a few nations who said, no, we don't want to be part of your free trade zone. We'll just be sort of independent nations. That's what those three organelles are about. So let's finish up our discussion of the membranous organelles by looking at these non-endomembrane system membranous organelles. The peroxisomes are kind of interesting. They are a digestive organelle, but they specialize in digesting fatty acids. That small little ball-shaped thing right there is supposed to be a peroxisome. So they're more or less round-shaped with a membrane on the outside. This is supposed to be a fatty acid molecule. And remember that fatty acids are the building blocks of a lot of the lipids, like triglyceride lipids and phospholipids. The fatty acid molecules can be used for energy by the cell, but first they have to be digested, and that's what the peroxisome does. It takes in fatty acids, and it also takes in oxygen. It uses the O2 molecule to digest the fatty acids down into smaller bits. which this is then available to the cell for energy. And I always thought that was kind of curious because the cell already has a digestive organelle. What's the major digestive organelle called? Lysosome, yeah. But for some reason, it has to have this specialized digestive organelle, the peroxisome, just to specialize in digesting fatty acids. And like I said, I'm not chemist enough to know exactly why it is. It could be that something about the structure of a fatty acid molecule requires different digestive enzymes and the use of oxygen to break it down, whereas proteins and carbohydrates, maybe those are best digested with different enzymes in an acidic environment. But for whatever reason, peroxisomes just specialize in digesting fatty acids and nothing else. All right, another membranous organelle that's not part of the endomembrane system is that yellow-looking one towards the bottom. That's called the mitochondria, and it is a double membrane organelle. So it has not one phospholipid bilayer, but two. Here's a review question. What other organelle we've talked about so far is a double membrane organelle? Nucleus. Yeah, remember the nucleus also has two phospholipid bilayers. So mitochondria does also. The outer membrane of the mitochondria is relatively smooth, but the inner membrane is highly convoluted in all sorts of hills and valleys. And the function of the mitochondria is to make ATP. ATP is this high energy nucleotide that the cell uses as its direct power source, meaning that any enzymes or other proteins inside the cell that need energy to carry out their function, those enzymes just grab an ATP that's floating around in the cytoplasm and get some of the energy out of it and to carry out their task. And so the cell needs to constantly be manufacturing ATPs to make sure that its proteins have enough energy to do their tasks. Well, that's what the mitochondria does. It is the ATP factory. And to manufacture that ATP, it typically uses glucose. So glucose molecules go into the mitochondria, and then using the energy of the glucose molecule, the ATPs are produced and just sort of spill out into the cytoplasm. And then those ATPs are there for the proteins that need them.